Now we'll move on to supervised classification. Just like the unsupervised classification, we have to first identify clusters of pixels, and the spectral statistics from those pixels can be used to then assign all of the other pixels in the image to one of those clusters based on its values. The difference is that with the supervised classification, we actually identify what those clusters should be. The benefits of this approach is that we end up with clusters which will ultimately give us classes in our thematic output that are meaningful to us. They're exactly what we're looking for because we have specifically trained the software to look for them. And it also helps prevent confusion with spectral classes because typically when you're identifying your training pixels or your training signatures for those clusters, you've identified very pure spectra or spectra that you are really confident are that surface feature you're looking for. And so they tend to have a little bit less heterogeneity or variability than the unsupervised clusters might end up having. But the disadvantage of this approach is that it really does require that you have a lot more knowledge of your area, that you are selecting pixels that you are confident of their classification on the ground. So it also often requires more field work if you're out collecting field points. And then even creating these signatures, creating these clusters that the algorithm then will run requires a little bit more upfront work. So it's not quite as easy or satisfying as running that ISO data and very quickly seeing an output thematic map. The other disadvantage of the supervised classification is that it really is only going to be looking for the clusters or the spectral signatures that you've provided to train the image. So if there are other surface features in the image, chances are they may end up unclassified, in other words, not really being assigned to any one of your clusters because they're so different. So you may end up missing classes that you might have been interested in and might have decided to keep out as a separate class if you had run an unsupervised classification. But really, I think this approach is great when you know exactly what you're looking for and you really want to identify and isolate those features. And just to give you an example, a lot of people right now want to have an ash map for all the ash trees, aside from just is it deciduous or is it conifer, because they want to be able to know where to look for emerald ash borer as it moves through the eastern US. So that would be a very good argument for running a supervised classification specifically to identify ash pixels with ash trees in them, and then you could say we really don't care about the classification for any of these other pixels. The process for the supervised classification looks really similar to what we did for the unsupervised ISO data algorithm, except we have this additional training step right up front. And this is where we're collecting and evaluating the separability for whatever spectral signatures we're going to use to create those initial clusters, right? To create those base statistics that all the other pixels in the image will be compared to. And then we use this training data to run another clustering algorithm. So very similar to what unsupervised does, it's going to start clustering our pixels, but this time it's using the training data that we have assigned for its statistics. Then we go through the same sort of signature analysis when we're evaluating the statistics and the separability of our final classes after all the pixels have been assigned to them. And if we're not happy with these statistics, we might come back and redo our training or redo our clustering until we're happy and we're convinced that the statistics are showing us we have good separability among our classes. When we're happy with it, we can apply the classification. And then again, remember that last and equally important step of even creating the map is running the accuracy assessment so that you know how good your map actually is and can interpret it appropriately. Since we've already talked about the clustering algorithms and evaluating statistics in the earlier video on unsupervised classification, let's spend our time focusing here on this training data, right? This first step that's unique to the supervised classification. So it could be as simple as going into your imagery or overlaying your imagery on air photos or on DOQs or some high resolution imagery and identifying the surface features that you know know you'd like to be able to classify and differentiating them from each other. So you really can start collecting training points just by using another image or the image that you have itself. You also could go into the field and actually collect points, and this is particularly useful when we are trying to map tree species like that ash example I gave you. You can't tell whether you have ash just by looking at an air photo, but if you're hiking around in the woods and you find a nice ash stand, you certainly could take a GPS location and then collect spectra from that once you get back into the lab. You can also gather training data from what we call spectral libraries. So there are quite a few online databases out there that contain spectra of different surface materials. These are usually collected in the lab, so it really is a pure spectra of whatever that object is. And you can simply go online and download these spectra. And there are really a lot of them for things like mineralogy, geology, vegetation types, and a lot of these are housed within federal government websites, so quite useful, but maybe not necessarily comparing apples to apples. So in other words, I find that it's usually best, if you can, to grab your training data from within the same image that you're trying to classify, because we know that there's lots of things going on in here. There's different illumination depending on when the image was collected, there's different atmospheric conditions, and if you're trying to compare the spectra in an image that has all of those additional sort of sources of noise to what a pure spectra would look like within a lab, they may not always look very similar. So it's better if you could find what you're looking for within your image to use that. Now sometimes that could just be a single pixel, so you may have been out in the field and found that stand that you wanted to use for calibration, and you could go in with a GPS location and pull just that individual pixel and use that one value for each band as your cluster. Now ideally you could collect other pixels that also have the similar spectral characteristics because they have a similar composition or similar characteristics on the ground. For sort of less distinct classifications where you're just trying to classify broad categories of, say, water versus agriculture versus forest, you could go in and just delineate. In other words, you could literally digitize a polygon within the imagery to identify what you want, and that's just by hand, and that's visual, and it could take all of the pixels that you've outlined with that polygon and use those as your cluster. But there's also a really fun seed tool right here that we can use where you can identify a pixel that you're absolutely sure of, and you can place that seed tool on that pixel and then actually grow pixels from that seed. And what the software does is it identifies all of the neighboring pixels that are still spectrally similar, and it clumps them all together into a larger polygon for you. So what's nice about that, instead of defining it yourself by hand, is that it's actually looking at all of the bands in your imagery, whereas if we're doing it visually, we can only see 3 bands at a time. So the seed growing tool is really a pretty nifty way to identify a larger number of pixels, so you're maybe getting a more robust spectral signature for your cluster. And there are lots of different thresholds that you can use for this seed growing tool. So you could be very conservative, and if you planted your seed, say, in the middle of that lake, you could say, I really only want the pixels that are almost exactly the same as my seed pixel. And then it might only grab a few pixels surrounding your seed. Or you could be a little bit more lenient with that choice and say, as long as it's somewhat similar, and you give it some statistical bounds. You can also give it bounds in terms of the number of pixels that you want it to grow. You can give it bounds in terms of how spectrally similar, so the statistics of how different or similar it has to be. So there are different settings that you can play with, and there is no set threshold that you should use for those settings. You really have to play with them and adjust them. Try out the seed tool and see if it grows to what you're trying to capture. Adjust it, maybe tweak those thresholds, and rerun it. Just like when we were collecting our GCPs for the geometric correction, we had these guidelines to be able to make sure we were getting good training sites. Well, we have similar guidelines when we're collecting training signatures for our supervised classification. Some of them are pretty obvious. So, for example, you'd like to make sure you have an adequate number of pixels so that you're really representing the natural variability within that surface feature type. I mean, all forests are not created equal, all agriculture is not created equal, all development is not created equal. Roofs aren't even, different roofing material, different asphalt. So you want to make sure that you have a sufficient number of different locations within your image, or at least pixels, even if they were from one location, enough pixels to represent the true variability in that surface feature. You'd like them to be relatively homogeneous though, so you want to capture the variability but not have them be so variable that so many pixels could all be classified to them just because the standard deviation is huge for that cluster, right? They have to be unique and separable from the other clusters. Again, just like with GCPs, you'd like to have them distributed throughout the scene, and that's because the atmospheric conditions or the illumination may be slightly different in different parts of your image, especially if you have images that cover a large geographic area. And so you want to make sure that if you're grabbing, now say for example, you're trying to grab spruce fir forests and you see them on the eastern side of a mountain, that you also have spruce fir pixels to train with from the western side of the mountain, because they will look different just because of the illumination angle. Obviously, you have to make sure that these are the right spots, that if you're pulling field data, you've got accurate geolocations. Some of those handhelds are not very good, so if you're not 100% sure, just don't use it. Now this one is where I think it's maybe not as obvious. If you are trying to grab training pixels, it's actually better to have fewer pixels, but pixels that you know are really just composed of the surface feature that you're looking for and are not mixed pixels. That's the best. So often you will see people grab training points, and let me give you an example. So for example, here is this lake. Rather than going and trying to get every single pixel in this lake and really going right along the edge where I might have some mixed pixels where the land and the water actually both cover the same pixel, I might just go for the center here and make sure that what I'm grabbing is water. And the same thing here, you can see that I've stayed well within the boundary of what that actual object looks like on the ground. If you don't have training data that represents really all of the possible classes of features you would expect to find in your image, so in other words, all the spectral variability, you're going to end up with a lot of pixels that are just unclassified or incorrectly classified. So it's useful to do an exercise of thinking about how many different classes you actually think there should be within your image, and trying to capture each of those with training data, right? And obviously this particularly includes anything that you're interested in classifying. It's all done in one fell swoop, so you want to make sure you have training data for everything that you're looking for. Now, don't be confused between this one saying you should have at least training sites per class and an adequate number of pixels. Really, the pixels could all be located from just one site. I could have 100 pixels from one site. What this is saying is that even if you only have one pixel at each site, try to make sure that your sites where you're gathering those pixels are dispersed and are spread out. And again, that way, hopefully, you're capturing the natural variability within that cluster. In lab this week, we'll be going over how to gather these training points and how to evaluate them. And for the most part, it is very similar to what we did with the unsupervised classification. But we also have some additional metrics that we can use for our supervised classification before we even run the clustering algorithms. We can simply look at our training data and the pixels we've collected for that training data and examine the histograms for each of these. Now remember, a histogram is showing you that for a given band and all the possible values for that band, how many pixels within each of your training classes have a given value. So the higher this peak, it means the more pixels had this particular value for this class. And there were no pixels that had a value less than this or greater than this for this yellow class. And each of the different classes, the different training cluster signatures we've collected, are in a different color here. And the idea is that we should be able to look at these and see if they overlap and how much they overlap. Because if we get a pixel value that falls somewhere on the edge, we're going to have a lot of confusion, or the algorithm is going to be confused about whether it really does belong to this one, the yellow one, or to the orange one, which also seems to overlap this same range. And that's why the standard deviation, in other words, the spread about this mean peak for your given cluster, is so important. Because if we see that, okay, well, if you have a value of, say, whatever this is, 40, it could be either the orange class or the yellow class. But if it's really far from the mean of the yellow class relative to that class's standard deviation, then the algorithm will know to assign it to the orange. But this is just another metric that we can use to say, are our training sites really separable? Are they distinct? And are they capturing enough variability? Ideally, each of your signatures would be a very narrow histogram, looking at sort of a normal distribution, that bell-shaped curve, and there really wouldn't be that much overlap in band values. But just inherently, some surface features are more variable than others. I'm going to guess that this is water right here. I mean, water typically looks like water. There's not a lot of variability unless you're in a region that has a lot of variability in, say, algae levels or pollutant loads or sediment loads. But other features, say, for example, agriculture, you have fields that are in different stages of growth. You have fields that are barren. You have fields that are growing different crops and might have slightly different spectral signatures. So some inherently are going to be a little bit more variable. Another few things you can look for when you're examining those spectral signatures in the histograms is, one, how many peaks do they have? So remember, you want that nice bell-shaped curve with one peak. If you have a bimodal distribution, which means there's actually two separate peaks, it means you probably have more than one actual surface feature that you're capturing within that training cluster, and you might want to go back and create two separate clusters for training. If you see a histogram that's very spiky, in other words, it has a whole bunch of little peaks, not one real obvious peak, it means that you really have a lot of variability in your data in that surface feature. And you may know as a scientist that that's normal and there's nothing that you can do about that, or you may realize that you might need to go back and clean up your training sites because you just have way too much variability in there. Maybe you have a lot of mixed pixels included in your training. If you see a really small histogram, remember the height of that histogram represents how many pixels have any given value for that band. And so if you see a really short histogram, or it's not even really a bell curve, it's short and it's also kind of spiky, it's probably telling you that you just don't have enough pixels within that entire training cluster and you need to go back and collect more. What we're really looking for is that nice normal distribution without a lot of overlap with the other bell curves, the other classes, as they're plotted on that histogram. There is another visual tool that most software packages give us, and that is ellipse plots, or they sometimes are called feature space objects. And what it will do is actually take each of our training clusters and it will plot them out in two-dimensional space. So you have to pick two different bands. It would be the two that you think might contain the most information. Typically, that would be your NIR band and your red band. And it will plot them out with their means and standard deviations so that you can see how those clusters lay out in space, in spectral space, for all of the pixels in your image. And again, what you're looking to make sure is happening is that you have all the classes you need to cover that full range of pixel values, right? So we don't have any parts of this sort of purpley sideways histogram that we're not representing, right? We have some class over each of those different areas. But the other thing that we want to do is see how much overlap there is between each of these ellipsoids. If we have different classes that overlap significantly, shrubs and grass, that's quite a bit of overlap. We might want to go back in and try to refine those training sets before we run the clustering algorithm. Ideally, each of these ellipses would be very small and distinct from the others, but it hardly ever works out like that. You almost always are going to have some ellipse that's overlapping with a whole bunch, and that really is especially a problem if your ellipse is very large, meaning that there was a lot of variability in that data set that you collected for training. And again, we could have known that by looking at the statistics as well, right? We can go into the statistics for each of these training clusters and look at the standard deviation. And remember that threshold. If you're below a standard deviation of 1, then you're probably not really capturing how much true variability there is in your surface feature. But once you get up to, say, 9, you probably have way too much variability to really be able to distinguish that from any of the other surface features. Another thing that you can be looking for again is, did you miss any areas where you have no cluster, no training cluster that its ellipse covers the pixels that you know you have within your entire image, right? This is telling you that you've missed some surface feature in the training clusters you've provided. And what's nice about most of our software packages is if you do find a location within your two-dimensional histogram where you do not have an ellipse, a training ellipse to classify, you can click on that area and it will bring you to those pixels within the image. And so you can look and see what that is and maybe create an additional training set. Also, just like we did in the unsupervised classification, we can evaluate the separability statistics for our training clusters, and we use that same threshold, right? If we have a separability that's less than 1600, then that tells us that we do not have a good distinction between two of those different training classes, and we might need to go back in and decide if they really are two distinct classes or if maybe they should be merged. So if you haven't figured it out by now, all of these classifications are iterative processes. So you might start with your training sites and collecting your spectral signatures for training, and then cluster your pixels, evaluate those statistics, and if they aren't good, or there's some that are good and some that aren't, you fix those and refine them, and you go back and you do it all over again, evaluate the statistics again, and you keep doing this until you're happy with your standard deviation for each of your different training clusters. You're happy with how the histograms look. You've examined the ellipse plots and they seem pretty good. You've looked at the separability data values to make sure that you have separability approaching 2000. And then there is one other piece that we can use that is in most software packages, and that's a contingency matrix. A contingency matrix is basically an accuracy assessment, but it's not a true accuracy assessment because it's not independent. All that it's going to do is go back through all of your training pixels. So these are the pixels that were already used to create the statistics for each training cluster, and it just pretends that those pixels were not a part of the cluster. It holds the cluster statistics the same, but it now goes and it looks at every pixel and it tries to classify them. And then it gives you output that's basically a matrix of what each of those pixels, which cluster they were truly in for training, and which cluster they are predicted to be in if the algorithm were run on those. And ideally, especially since these are your training pixels, and they should be pretty pure in terms of whatever the surface feature actually is, you should have accuracy that's near 100%. So in other words, all the pixels when they're classified should fall back into the clusters that they originally helped to create. So you know you have a good signature for training if your accuracy is near 100. So the way this matrix works is that you have all of your classes across the top as different columns and the same classes down the rows, and you'd like to see that every pixel that truly was the AG1 class becomes classified as the AG1 class. And so here you can see that there were 100 pixels that were truly AG1 and were classified as AG1, right? And so we don't see any other pixels that were classified as AG1 or that we've missed. So this is a good signature. But on the other hand, if we come over here to this forest 1, we can see that there were 407 pixels that were accurately classified for forest 1, but that there were 17 pixels that instead of being classified as forest 1, which they really were, they were classified as forest 2. And we see the same confusion over here. So in other words, there were 286 pixels of forest 2 that were accurately classified as forest 2, but there were 14 that were misclassified as forest 1. So again, this is when you have to come back to your objectives. If you're just trying to differentiate forest from ag, then this isn't a problem. But if you're trying to identify different types of forest, maybe evergreen or conifer, or different common forest assemblages, this may be of concern and would indicate that you need to go back and it's tempting to think that we're done once we're happy with our training clusters, but actually we've only really made it to the part where we can actually run the classification. So we've collected all of this training data and we've evaluated the clusters in a bunch of different ways. And once we're happy with those, we need to now go back and run every pixel in the image and figure out which of our training clusters it most likely belongs to. So this is where we're actually classifying the data, classifying the image. And there are lots of different ways that have been developed to figure out which different cluster every single pixel belongs to. And we're going to go over some of the most common ones, but basically these are just different ways to examine the statistics. They're different statistical rules to decide which class each pixel should go into. The simplest approach is the parallel piped approach, also called a box classifier. And it's called a box classifier because essentially what it does is it takes the minimum and the maximum value for each of your different clusters in each of the different bands. And it then looks at all the other pixels. So these in blue, these were my training pixels, right? Are all the pixels that I collected from my spectral signature for each cluster. And what it does is it looks at every other pixel in my image, which is going to fall somewhere else in this n-dimensional space. And for some it'll be easy. They'll fall right within this minimum to maximum value bounding box. But for others, there'll be pixels somewhere in the middle. And it's really just assigned, these pixels are assigned to whichever box they happen to be closest to. So in that sense, it's sort of like a nearest neighbor strategy, which again we saw in our geocorrections lab, where basically whichever box is nearest to you, you're just going to be assigned to that. It does have some problems though if you have a pixel that happens to be equidistant from multiple boxes, and in that case you would have unclassified data or pixels that were not able to be assigned to any particular cluster. Another common classifier is called the minimum distance classifier, and this one does use statistics, but it only uses the mean of each cluster, right? So it's not looking at the full distribution. The standard deviation or variability within that histogram isn't included. It's literally just the mean. And so just like with the parallel piped, it's going to assign each pixel to its nearest cluster, but instead of it being based on the boundary box of that cluster, it's which cluster mean it's closest to. So if you want to think about what that looks like, now instead of having these boxes drawn around each of our training clusters, we just really have the mean, this little pink X in here. And we're looking to see which of these pink X's, which of these cluster means each pixel in the image is nearest, and that's the cluster it's assigned to. One of the more common and more complicated classification algorithms is a maximum likelihood classification. So this one actually does use both the mean and the standard deviation. So in other words, it's looking at your entire histogram, not just where the mean for the different spectral values are, but how much variability there is within that entire cluster. And so what it actually does is a statistical technique where it calculates a probability that every pixel is assigned to a given class based on that mean and the amount of variability. And so it's going to assign each pixel in the image to whichever class it has the highest probability of belonging to. And for those of you who've taken a basic statistics course, or certainly my stats course, you'll find these histograms, these normal distribution curves, very familiar. And really what it does is it looks at the mean for the class, but it also looks at the standard deviation. And again, this is in multi-dimensional space, so it's a little bit more complex than what's being presented here. But essentially, the farther you fall from that mean, not in terms of actual DN values, but in terms of standard deviations for that training, the less likely it is that you belong to that training cluster. So this really is just using good old-fashioned statistics to assign a probability that a given pixel will fall within each cluster. These may seem like very academic distinctions between classification algorithms, but they really do result in different output. So what you see with a minimum distance classifier may look very different than what you see with the maximum likelihood classifier, because again, this one is only looking at mins and means and basically drawing a bounding box around each cluster. But for this one, it's using the probabilities. So notice, for example, how this bare ground that you have mapped in the maximum likelihood classifier is almost entirely absent in that minimum distance classifier, and instead ends up being lumped into the urban classification. So sometimes you can decide exactly which one you want to do ahead of time because you've looked at your histograms and you know how variable they are. Maybe if they're all equally variable, you don't really need a maximum likelihood classification, because just looking at the minimum distance may be sufficient because the amount of variability in each of those training clusters is the same. But other times you may actually have to run them in a couple of different ways to see which one works best. Sometimes it really is just trial and error and try to refine those training clusters. I think we'll leave it there for this week, and we'll spend some more time next week talking about accuracy assessment, and then also some post-classification processing that we can do to improve our final classifications, or at least to improve the accuracy of those classifications so that we're not putting out false data. But I guess the take-home message is to remember that there are lots of different applications. There's lots of different objectives when we're classifying imagery. Sometimes you really do just want broad scale, gross differences between really obvious classes. And it depends also on what type of imagery you're using. I mean, look at the pixel size for this, right? But other times you're interested in much finer scale differences to identify smaller objects or objects that are more distinct, for example, comparing a vineyard to an orchard. And this might be an example of a time you'd want to use a supervised classification, or maybe even one of the more advanced classifications that we'll also go over in the coming weeks. And in lab, we'll practice some of these different techniques with your downloaded imagery, your Landsat imagery. So make sure you know where that is and you're ready to work on that come Tuesday. Until then.